Good morning, good afternoon, everyone. My name is Mark Garner with Agilent Technologies, and I'm really delighted to be here today for Dr. Qing Wang's seminar. Dr. Wang is CEO of Complete Omics. And, you know, a lot of us have been doing or talking about protein quant, peptide quant by LC-MS for a long time. But I think what's been missing, we've done a lot of good technical work, but what's been missing is that killer app, that really clinically relevant application that drives the field forward. And I think you'll agree that Dr. Wang is going to talk to us about exactly that really clinically relevant killer app. The title of his talk is Hitting the Achilles Heel of Cancers with Confidence, Robust Validation of Neoantigens. And with that, I will turn it over to Dr. Wang.
Thank you so much for the introduction, and thank you for organizing this great event to allow us to present our work. So the topic of today's talk is about hitting the Achilles heel of cancers with confidence, robust validation of neoantigens. And again, my name is Qing Wang. I'm the founder and CEO of Complete Omics. So I will start by briefly introduce what the human leukocyte antigen system is. The human leukocyte antigen system, or HLA system, is encoded by the MHC gene complex in humans. These cell surface proteins are responsible for the regulation of the immune system. And the HLA gene complex resides on a 3 megabyte stretch within the chromosome 6. And HLA genes are highly polymorphic, which means that they have many alleles, allowing them to fine-tune the adaptive immune system. So there are mainly two main systems for HLA molecules. So type 1 HLA corresponds to MHC class 1, and type 1 HLA present peptides from inside the cell. For example, if the cell is infected by a virus, the HLA system brings the fragment of the virus to the surface of the cell so that the cell can be destroyed by the immune system. These peptides are typically 9 to 12 amino acids long and are produced from digestive proteins that are broken down in the proteasome inside the cell. Antigens presented by MHC class 1 attract T lymphocytes called killer T cells that destroy those cells. And type 2 HLA corresponds to MHC class 2. Type 2 HLA present antigens from outside of the cell to T lymphocytes. These antigens stimulate the multiplication of T helper cells, also called CD4 positive T cells, which in turn stimulates antibody-producing B cells to produce antibodies to that specific antigen. Self-antigens are suppressed by regulatory T cells. And the peptides that are presented by type 2 HLA molecules are typically 12 to 25 amino acids long. So the major topic of today is about neoantigens. So neoantigens are newly formed antigens that have not been previously recognized by immune systems. So we typically call the tumor neoantigens a name called MANA. It stands for mutation-associated neoantigens. MANAs can be presented on tumor cell surface and can be recognized by T cells. And which is also, neoantigens is also the foundation of all the recent cancer immunology therapeutics methods, and also those immune checkpoint inhibitors such as anti-PD-1 antibodies are dependent on the effective recognition of MANAs. But despite the importance of this group of molecules, it is very difficult to reliably detect and quantify neoantigens. therefore to validate their potential of being a drug target in cancer therapeutics field. So we have recently developed a technology which we call MANA-SRM. Basically is a technology that could allow us to enrich specifically the MHC molecules from the cell lysis, and then to use the neoantigens from the molecule, and then after purification and fractionation, so we will be able to detect those molecules and quantify their abundance. And this technology is built upon our previous development of two actually major technology. One is called MT-SRM, which stands for mutation targeted SRM, and another one is called SAFE-SRM, which is our recent development in the paper published in 2017. So the MT-SRM is basically a method to enrich mutant proteins from a variety of different samples, and then followed by the mass spec detection of the mutant peptides. So in this technology, we actually used the very commercially available antibodies to enrich the proteins that have a mutation on itself. Compared with using mutation-specific antibodies, this approach is very affordable because you can virtually use all the antibodies that are targeting the wild type, most of the wild type versions of the proteins, use that as an enrichment method. And then you allow the mass spec to detect the mutant proteins, mutant alleles. Basically is to combine the enrichment from the very commercially available antibody with the ultra high resolution of the mass spec to detect the mutant peptides. And also another part of this another important part of the MANA platform is called SAFE-SRM, which is another development that we recently published in 2017-18. So about a method that could allow us to detect ultra low amount of peptides from a variety of different samples, mainly plasma samples. So by combining these two technologies together, with further optimizations, we developed this MANA-SRM detection method. So if you think about the detection of neoantigens, there are really just three different difficulties that you want to solve before you actually make a major improvement. The first one is how can you optimize the enrichment procedure that you can enrich HLA molecules and together with the neoantigen peptides, but not other lysate part, not other fragments of the cells. And also, how can you prevent the antibodies from falling into your samples, therefore to block your detection sensitivity? And the second big question is, how can you remove the contaminations from the IP systems that to boost up your detection sensitivity? And the third question, which is actually very unique to neoantigen detection, is how can you stabilize all the peptides that have chemically unstable residues? So those are the three technical difficulties that we have solved when we are developing this MANA-SRM technology. So we first optimized a set of different IP-based enrichment methods and compared their efficiency and compared to their cleanness of the enrichment. So we first tried different cross-linkers, and the results shown here is only the part that actually the final result that we optimized. We figured that using DMP at 25 millimolar to trace the antibody with the beads together with the protein G beads give us the best bending of the signal, give us the strongest signal of the from the antibody enrichment. And also we tested different IP systems, and we first used our previous IP systems that composed of a Reaper buffer and some modifications, which I listed the reference here on the right side. And we compared that with another different protocol that claimed to be the best for the neoantigen enrichment. And also we compared it with a different protocol that claimed to enrich the cell membrane proteins. And we found that our previous method was able to enrich MHC molecules at the highest abundance. And then another step is to see what are the best elution buffers that could elute your neoantigens from the beads, but still keep the antibodies, keep all the small fragments of the antibodies away from your elution. So we found that using acetic acid at 20% give us the strongest signal across the different alleles of this HLA molecules. So, and also we quantify the abundance of the monomers, and we can see that the whole method can allow the enrichment of as high as 2 microgram monomers in 35 million SW480 cells, which is roughly can be translated into 800,000 HLA molecules per cell. This number means that our method is able to enrich all possible HLA molecules from a single cell. And during the sample preparation, we do the QC of every step. So basically, for every single step of the procedure, we save a little bit of the sample and analyze the sample loss and the signal suppression. And we found that there is a significant amount of PEG derivatives that are present in our IP immunoprecipitated samples, and for showing those signature peaks. And then we try a variety of different methods, and we found that using a Helix ST-based cartridge that give us the best cleanup efficiency that could allow us to keep the peptides but remove all the polymers. So the method with the 3 previous optimizations, we established the method MANA-SRM, and then we tested how can we further solve the issue of peptide detection. So in SRM field, when you want to detect a peptide, you actually are able to choose which peptides you think are the best to use as a target for each protein. Therefore, you, as a guideline, when you design those peptides, you actually want to avoid some chemically unstable peptides, such as the ones with cysteine, such as the ones with methionine, because you want to increase, first of all, you want to increase your sensitivity by seeing most of the peptide sequence that you want. For example, if a peptide sequence has 10 copies in your sample, and it has a methionine in it, maybe there are, during the sample preparation, the methionine will gradually be oxidized, and if you don't take previous precautions. And in the final detection, there may be only 2 or 3 methionines that are at their native reduced form, and all other like 8 or 7 methionines are all oxidized. Therefore, you actually dilute your detection and making your sensitivity drop. So in order to solve those issues, people develop many different ways, such as when we do SRM detection, people usually just avoid selecting the peptides that with cysteine or methionine or other like other residues that are subject to chemical changes. But for neoantigen detection, you really don't have a lot of options, because the neoantigen peptides, by definition, needs to be the peptide that's flanking the mutation site. And the mutation site, you don't really have a lot of options. For example, KRAS mutations, the TP53 hotspot mutations, those are pretty fixed sites. So which leaves you most of the time no other good choice to switch between different sequences that you want to detect. So in order to make sure that all the chemically unstable peptides are actually detectable, we develop another approach, which is called dual reduction. This approach includes reducing reduction event at high pH with a DTT. And then before we send the sample to mass spec detection, we reduce the sample again at low pH with TSAP. And through this procedure, we can see a significant amount of boost for the peptides abundance after treated with DTT and TSAP. I highlighted on this several of the dramatic values. Those are the fold change data showing the amount that we can gain after the DRED dual reduction treatment. So with that, we have established the whole procedure, including the optimized IP system and also a cleanup procedure to remove all the detergent contaminations from the sample, and also peptide preserving procedure to keep all the peptides with chemically unstable residues to be at their reduced format. And with those three improvements, we built the MANA-SRM pipeline, and we evaluated the recovery ratio of neoantigen quantifications. And we can see from this table that the average recovery ratio that we can observe from this pipeline is above 70%, which is dramatically improved from the previous only 1 to 3% by previous publications. With this enrichment, this kind of enrichment efficiency and the recovery ratio, we are able to see really little tiny amount of the sample, and we are able to see neoantigens that are present at the cell surface at very low copy numbers. And here we present the data of the quantifications of neoantigens from our MANA-SRM pipeline. So you can see that we first perform the transfection procedures to transfect HLA allele together with vector that express the mutant gene, mutant neoantigens into the CO7 cells. And we measured abundance of the presentation of those neoantigens at the single per cell level. And the most right side column shows the copy numbers per cell. And with those information, we further quantify the neoantigen presentation from a variety of different endogenously mutated cells, and such as the lung cancer cell lines and a couple of other cell lines that I listed here, and on the left side. So from those data, we can see that the sensitivity of our technology is able to quantify down to 1 copy per cell, and which is a dramatic improvement over previous methods. So I would briefly talk about the importance of the work that we did. So in recent years, there have been a big effort in the whole field to try to optimize the peptidomics data that enriched from these HLA molecules, and there have been major publications which are listed on the right side of the slide, showing a potential of neoantigen targeted therapeutics methods. So we are trying to contribute to this entire campaign by coming up with this MANA-SRM method, which allowed us to contribute to two recent publications in March. One is on Sense, and the other one is on Sense Immunology. With our contribution to the project led by Dr. Bert Vogelstein in Johns Hopkins, we were able to help him build bispecific antibodies targeting a common TP53 mutation, and also another bispecific antibodies targeting a very common KRAS mutation. With that, we are currently working with 6 pharmaceutical companies to validate their neoantigen target for their cancer therapeutics approaches. And on top of this this seminar, we actually are just submitting a paper for review describing a new pipeline that can directly handle patient tumor samples. So the current technology we have is able to analyze neoantigens from down to 50 milligrams of a solid tumor. So which we think is a big improvement that could push the whole neoantigen detection technology into direct patient care. Because we are very dedicated to push our technology to the most sensitive level, because we know that the more sensitive our technology can be, the smaller tumor tissue that we can handle, the more patients can benefit from this, from the personalized neoantigen-based therapeutics. Previously, it has been a big difficulty to directly handle patient tumor samples to evaluate their neoantigens. Usually, you will see the publications with tumor hundreds of milligram level, and but those are representing only a small part of the cancer patients, which are relatively middle to late stages. The patients who actually can benefit most from personalized therapeutics are actually the early diagnosed, are actually the ones that can be early diagnosed. So we built up this new pipeline that could help to include more patients into this potential personalized therapeutics approach. So and finally, I want to demonstrate this, show this to you guys, that we are welcoming any members to join us with as a team member, and also we are currently offering neoantigen validation services to the scientific community and also to the pharmaceutical companies. And if you want to discuss with us about the collaboration, please send an email to info@completeomics.com. And with that, I want to thank, first of all, Dr. Bert Vogelstein, who has been my mentor over the years, and he has been very supportive to my company after we were founded, and we are still collaborating on a variety of different projects. And also, I want to thank the Complete Omics team with our fancy team members that make all the improvement of our technologies. And the third, I want to thank Agilent Technologies to organize the event and also to give us constant support to our research for with their platforms, with their technical support, and their scientists works with us and answer our questions all the time. And with that, I want to thank you for being here and for attending this seminar. And I would want to and with that, I want to start to answer some questions if you have. And thank you so much for being here.
Thank you, Dr. Wang. That was great. And we have had a number of questions come in. I'll start. First one, someone was asking how long the entire procedure takes and about the cost of materials for a single assay.
I'm very happy to answer the question. The whole procedure, starting from dry cell palette, will take about 6 to 7 consecutive days to finish, and it will cost roughly $5,000 just for the experimental materials to perform one assay. And the most costly part will be the antibody and the magnetic beads. Each of them will cost about $2,000 to get. We need usually a high milligram levels of antibodies and more than 10 mils of the magnetic beads. Those are the most costly part of the experiment.
Okay. Okay, great. And actually, less detailed question about how are you planning on targeting the cells with MANA-peptides?
I mean, there are so many different ways of when you have the neoantigen information. There are so many different ways that can be adopted to design a personalized therapeutic methods. People have tried using mRNA vaccine, to encode those neoantigens, and also people tried using just directly injecting neoantigen peptides to human body. And also, just like what we recently published with Dr. Bert Vogelstein, that we used bispecific antibodies to achieve the therapeutic effect.
Okay, excellent, excellent. And those were the science and the science immunology papers, correct?
Right. Yes, correct.
Yeah. Okay, great. Thanks. Do you ever see PTMs on the MANA-peptides?
That's a great question. We haven't seen design approach to just see the PTM. So far, we have been focusing only on missense, point mutations. There are still a lot of questions need to be answered, such as which the most frequently mutated human cancer driver genes, which mutations actually can be presented on cell surface. I believe that next step, after we figure out a complete list of the frequent neoantigens, that we should move forward into those PTMs that can be presented on neoantigen peptides.
Okay, excellent. And one fairly detailed technical question, 2 parts. Why do you need double reduction? Do you detect artificial methionine oxidation without the reducing agent?
So double reduction, the reason is there are 2 steps that will actually, in the whole procedure, that will actually causing your sample to lose sensitivity. The first step, imagine that if you have a peptide, I mean, it's like the same sequence, but with brothers and sisters who are modified at different positions, then you will lose the sensitivity because let's say you have 10 peptides that was supposed to be detected. Let's say you have a sensitivity of detecting 5 peptides, but you actually have 10 copies of the peptide. Yes, you can detect it. But if those 10 peptides are, becoming 10 different derivatives, then you won't be able to detect any one of them because they become just one copy, which falls below your instrument detectability. So we did a high pH fractionation, which is a step that we purify the samples, and we believe that if there are different modifications during the fractionation, the same peptide will be fractionated into different positions, different fractions of the sample. So therefore, we actually will result in reduced amount for each individual fraction. Therefore, we hope to preserve the peptide that focus them into one fraction. So therefore, we adopted a DGT reduction at high pH that is more in line with the high pH fractionation. And then in the mass spec analysis, it is low pH. So it still has the same problem if we don't keep the peptides well. Different different format of the same sequence will actually become different peaks in the low online LCMS. So therefore, we need to further reduce them using the low pH reduction buffer, which is TSAP. With these two approaches, we are able to basically refocus all the separated peaks into one single peak for each individual peptide sequence. Therefore, we can boost up the sensitivity of the detection.
Okay, great. And for determining copy number, so the number of copies of the neoantigen per cell, how do you do that?
So first of all, we are able to quantify the neoantigen copy numbers from our sample, and we know the starting cell number of our sample. So then we divide these two values to calculate the copy numbers per single cell. It's an average level.
Okay, great. And a question came in, are the antibodies specific to the mutation sites?
Oh, no. The antibodies are not specific to mutation sites. We use a universal HLA antibody, W632 clone, to enrich the HLA type 1 molecules. It's not neoantigen specific. It will enrich everything it will enrich all the HLA type 1 alleles molecules, and with all the antigens and neoantigens are presented on the molecule.
Okay, makes sense. And how do you search for the neoantigen? Is there a database, or are you doing de novo sequencing?
Yeah. So this is basically a targeted detection. So it's, I understand your question. So what we believe about multi-omics is basically we need to know the genomic information, and with that we can more wisely design approach to make our detection more sensitive. So therefore, the presentation today, the technology we are talking about is a targeted detection approach where we know those mutations through genomic sequencing, and we can come up with a list of possible neoantigens that that could potentially presented on the cell surface. So, because the mutation site is known, and also the length of the possible neoantigens is from 9 to 12. I mean, some of them are 8 amino acids, but most of them will be 9 to 12 amino acids. You actually can exhaust all possible neoantigen sequences for any single point mutations. We also do profiling approach to just share the same sample preparation method. It's just we are using the Orbitrap rather than using a triple quad. Through the Orbitrap, we can get a complete profile of all the peptides, MHC peptidomics data, and we can do that. But from our experience, most of the data you get from those profiling approaches have nothing to do with mutations and have nothing to do with neoantigens. So therefore, the high abundance unrelated peptides are actually blocking your sensitivity from seeing those true valuable neoantigens. That's why and also right now, it is so readily available to sequence a tumor sample, and you need only 5% of the, compared with the tumor tissue you need for proteomics approach, you probably need only less than 5% of tumor tissue there to just get a complete coverage of the whole cancer genome. Therefore, you get a lot of genetic information in hand. With that information, you design a more targeted approach. We believe this is more sensitive and is better leverage the knowledge we know about cancer genomics and proteomics.
Right. Yeah, and we all know how precious those samples are, and you don't get a lot. Yeah. Actually, an interesting question came in if you think that this detection of neoantigens would work with FFPE samples.
That's a great question. We haven't tried FFPE samples. I think I would imagine that there could be some issue with the chemical modifications that are potentially happening on the peptides, on the neoantigen peptides. But we are happy to proceed and to continue to do some tests with FFPE samples if we have the resource.
Okay. See, one last question came in about the triple quad you're using.
Yeah, we have been using Agilent triple quad mass spectrometers, and the recent ones that we are using is the 6495B and C, and so those are the platforms that we have been using.
Okay. Okay. If anyone else has any questions, please enter them in the chat. Another one just came in. Do you think neoantigens will have a better clinical performance than genomic or mRNA molecules?
So it's a great question. So the very famous the COVID-19 vaccine makers, they have been starting with actually cancer neoantigen targeted mRNA vaccines before, and so there are a variety of different reasons. The results have not been extremely good. So, but I have seen over the past year with the widely used, widely optimized manufacturing procedures for the COVID-19 vaccination, mRNA vaccines, there are a lot of technical difficulties are being solved towards using mRNA vaccines to other diseases. So I would imagine that within several years, there should be a major improvement in directly using the mRNA vaccines for cancer vaccinations. So those, but one of the fundamental question is which sequences are actually presented there could serve as your target for the vaccination. Eventually, when the manufacturing method becomes very mature, it will be more of a market that for every individual patient, you need to know exactly what your mutation-specific neoantigens are presented. So that's why we develop further into all those technologies, and we hope one day we can serve as the first line of the patient's personalized therapeutics establishment to before the patient pays a ton load of money for their treatment. We can tell them exactly this is your target and you need to do this target, and don't use this one, don't use this drug, but use this target, this antibody. Or so that's basically is what we can foresee in the future, in the near future. Yeah.
Excellent. Another question. For people who are using predictive algorithms to predict neoantigens, have you been able to evaluate the efficacy of the exome transcriptome analyses by bioinformatics, basically to predict the neoantigens that you see by your technology?
Yeah, that's a great question. A lot of people have been working with prediction algorithms, and from the slide, there is a table showing you the copy numbers per cell, and you can search the sequences through MHC, net MHC, and you will get a general, you will see the result that the ones that actually presented the really high copy numbers doesn't correlate to the binding affinity that is predicted from those algorithms, and this is what we are. I think the prediction is good in terms of giving you a general idea about what could be there, and but I would buy in more on this technology improvement to finally exhaust all possible neoantigens on the cell surface to directly see the sequences, because we have seen the largely uncorrelated data from what we can see from the cell surface compared with the prediction. I think prediction is a good start point to give you some general, maybe good candidates to start validate. But it's great. It's much better to have the data directly for the direct observance.
And if I can combine a couple of questions, basically sort of what are the barriers to actually getting this into the clinic? And combining another question with that, do you think you have the sensitivity now? Is it just a question of I don't want to say validation because that can be a loaded word. How do you get from here to there, if you will?
Yeah, so it has been a big question mark for putting mass spectrometry-based analysis to directly patient care. But I believe this is neoantigen validation is a good starting point because there is a rigid need for the patients to have a personalized therapeutics methods, to have therapeutic approaches. And I think the most difficulty part is the reproducibility and the sensitivity. How can we come up with a pipeline that can be reproducible regardless of the operator's experience and regardless where this machine is located, but still see the same result across different hospitals throughout the whole world? That's a big question. The second question is how can we see the things that are at extremely low abundance? How can we ensure our sensitivity of detection across different institutions? So that's the major improvement that we are making, which we already submitted a paper, is that we are making an actually independent instrument that is able to process all the samples. It turns from a 6-day or 7-day approach to a procedure that lasts for only 4 hours. And with that, we believe that it will dramatically improve the accessibility of hospitals to the directly analysis of patient tumor samples. So that's basically what we are doing, and we hope to solve the reproducibility and sensitivity issues.
Yeah, excellent, and certainly automation, as much as throughput, is about reproducibility and precision. Yeah, great. Another interesting question: will tumor suppressor gene mutations produce neoantigens?
So tumors, we haven't specifically isolated, different mutations, and I'm sure tumor-specific mutations will produce neoantigens on the cell surface. So yes.
Sure. So question asked, do you see a correlation between intracellular neoantigen peptide concentration and the number presented in the MHC complex on the cell surface?
So we didn't specifically isolate the intracellular neoantigen peptide, but from numerous previous reports, it has been shown that the intracellular neoantigen peptide are less than 5% of the entire neoantigen pool. So we are more focused on the cell surface. Yeah.
Yeah. Okay. Excellent. Excellent. And then one last question, and this is a bit of prediction. What do you see as being the ultimate clinical detection assay? Is it SRM or? And that sort of goes back to what you said earlier about mass spec in the clinical lab.
Yeah. So. I understand. So I think the clinical assays will be the difficulties will actually be the sensitivity and the reproducibility. So it's not very much mutation variable, mutation specific. So I think let's say we believe that within 1 or 2 years. There should be a relatively comprehensive list of all the major driver genes mutations that can be potentially presented. So with that information, we could establish targeted detection approaches and with further improvement on the sensitivity for each particular patient that has a matched mutation. And I think it's possible to directly use some antibodies that are recognizing the neoantigens on cell surface, directly use those antibodies to do diagnostics. I think it's possible, but it will depend on established knowledge of a good list of very solid presenters and a good the establishment of a good library of antibodies that are very specifically reacting to those neoantigen sequences. Those will actually later, I think this will take years, several years to develop. But so far, we have been focusing on polishing our technology to make it more and more sensitive. Therefore, we can achieve that final solution, faster and faster. So, yeah.
Okay, excellent. And I think with that, we will wrap it up. And first and foremost, I want to thank you, Dr. Wang, for a fascinating and very stimulating presentation. I want to thank the audience. This was a great discussion, far more than we had hoped, really insightful questions. Remind everyone that this presentation will be on the on-demand site on LabRoots. You can continue to submit questions to the on-demand, and we will try and get those answered. And I think with that, we can wrap up, and I will thank you all for attending and hope that you enjoy your days.